The overall goal of the following experiment is to utilize a new and rapid signal amplification method to detect and characterize influenza viral subtypes on the flu-chip low-density microarray platform. This is achieved by incorporating biotinylated dNTPs and using primers with a 5' phosphoryl end to amplify the matrix gene segment and a non-structural gene segment from influenza RNA extracts. PCR is followed by enzymatic digestion and fragmentation to generate short, single-stranded, biotinylated, and amplified viral product, which is then hybridized to the array. After hybridization, ampliPHOX Detection uses photopolymerization to grow large polymer spots where hybridization occurs, and differential hybridization patterns are used to detect influenza viral subtypes.
The main advantage of this technique over existing methods, like standard fluorescence detection, is that it's rather inexpensive and provides a quick result, visible to the naked eye with similar analytical sensitivity to fluorescence.
Though this method can provide insight into the characterization of influenza viruses using the flu chip, because it's a universal detection method, ampliPHOX can also be applied to a wide variety of other systems, such as other DNA and protein-based low-density microarrays. In theory, any biotinylated product should be able to be detected using ampliPHOX. Demonstrating the procedure will be Amber Taylor, an associate scientist, and Kevin Moulton, a research assistant here at InDevR, who have both been central in the development of the ampliPHOX instrument and detection chemistry over the last several years.
Extract viral RNA from 200 microliters of viral isolate using a viral RNA extraction kit and the manufacturer's protocol. Elute